As I was saying previously, for everybody online and in person, Dr. Zhang and I turn out to be distant academic relatives. She's my academic aunt. I'm her academic, nephew? Her graduate advisor was the father of my graduate advisor. So again, it's a small world out there. Be nice to each other. Dr. Zhang comes to us from the University of Pennsylvania, where she's a tenured professor. After doing her dissertation, postdoctoral work, and her junior faculty start at Stanford University in statistics. Dr. Zhang has expanded the frontiers of the field and methods work in diverse areas, including spatial transcriptomics, immune profiling, and subclonal profiling, phylogeny reconstruction in cancer evolution. So if everybody would please give a warm welcome to Dr. Nancy Zhang for our final keynote of Bioconductor 2023. Thank you, Dr. Zhang.
Thank you, Tim, for the introduction, and thank you to all the organizers of this conference. It's really an honor to speak here. I'm pretty sure I've used the tools of many of the people in this room. It's the lifeblood of our work, Bioconductor and other open source software. And it's certainly our lab's one of our main goals to develop code that other people can use and follow up on. So yeah, I cherish this opportunity to share with you my work. So like many people in this room, my lab has been trying to grapple with all the opportunities that single cell and now spatial transcriptomic data can offer us. And in this talk, I want to share with you some of our efforts in this direction. And we're, in Dana-Farber, so I think, there's plenty of cancer genomics experts here as well. So I'm going to jump directly into the example. So this running example will be what I use to sort of tell the story of our methods development. So this is a H&E slide of a colorectal metastasis to the liver. Okay, so if you were a pathologist, you'll be looking at this and say, okay, the cancer is right there on the left side, the dark purplish region, right? And if in cancer, one of the things that why it's interesting to me, it is a manifestation of evolution within our tissues, right? So our understanding of cancer is that there are driver mutations that bring a normal cell to make it malignant, right? And then that leads to the growth of a primary tumor. And then there's a lot of heterogeneity among these cells. There's selection. And then if you were to apply a drug, maybe it will kill a portion of the cells, not all of it. And unfortunately, there might be ramifications down the road like metastases, right? So in our study of cancer, this intra-tissue heterogeneity is very important, which is why it is so exciting now. We can look at it through single cell and spatial methods. So I was actually working with these kind of cancer type of problems back in the 2000s, and now this is sort of like a fossil, right? In the grand scheme of how fast things evolve. So when I first worked with cancer genomic problems, we were doing bulk microarrays, right? And DNA and RNA profiling. For example, here you see a profile of a DNA copy number of cancer, right? So you want to find regions where there's gains and losses. And we have found since over the 20 years that these really matter in terms of driving as one of the driving forces of the disease. And certainly, as a prognostic factor as well. So about 8 years ago, right? So that's when a lot of single cell data started coming out, right? And then since then, we have used single cell profiling to understand, what is the heterogeneity of tumor cells. I mean, at least we're starting to understand that, right? And then we've discovered many things like cancer stemness, the stems of cells, and how epigenetic and transcriptomic changes that in fact added 2 hallmarks to our understanding of cancer. So some of this talk will go into the latest spatial transcriptomic profiling data. And it sort of overcomes this basic limitation of single cell profiling, where seeing single cell profiling, we dissociate the tissue, and therefore we don't know the location of the cells relative to each other in the tissue, right? So spatial transcriptomic profiling gives us that, but there are limitations, for example, spatial resolution. Right, so that leads to new analysis challenges and new challenges in the development of code and software for manipulating and understanding this data. So I'm going to touch upon this sort of our progression from our understanding of intratumor heterogeneity from bulk to single cell to spatial. Okay, so for example, for this cancer, specific cancer sample, one of the first things we might want to know is just how much diversity is present among the cancer cells, right? How many subclones there are? Is there subclones? And what are the mutations that mark these subclones, right? So with single cell data and spatial data, we're at the level where we can, I can tell you, we can, with some precision, get useful estimates of copy number per cell, copy number profile per cell, and or per spatial spot, because we're using VCM data. And based on that, we can do subclone detection. And I will show you some of the latest insights we've drawn on that front. But I can admit upfront that when you do subclone detection and copy number-based subclone detection of tumor cells, we're at the level where we can do it fairly confidently in single-cell DNA and single-cell ATAC sequencing data. And that was one of some earlier papers were on this. But then when you do it using, when you are trying to do subclone detection using RNA data, there's a lot of challenges. And a lot of spatial transcriptomic data is RNA level. So I will touch upon some of the computational challenges on that front. And then, we want to get to the really exciting stuff that spatial transcriptomic data offers us, which is now that you know how, the intrinsic genetic heterogeneity of this tumor, how do we tease apart how the different subclones interact with this environment? So sort of genetic cross-environment effects, right? So that will allow us to maybe ask questions in the future, like whether the cancer develops different strategies in its invasion or is in its escape of the treatment, right? So these are down the road, but at the beginning is we want to understand how it interacts with microenvironment. So I will tell you about some of our efforts along this front, including a model that we developed for niche differential expression analysis. Okay, so let's first look at the intratumor heterogeneity aspect. So this is based on two works. They're both posted. One is using single-cell DNA and ATAC. That's the first paper, and then the second paper is on using RNA level measurements. And these were led by two talented students, Jiyun Wu and Jiazhong Ren at Penn. Okay, so how much diversity is there in serial karyotype between tumor cells? So I want to just highlight some of the previous works that led to our work and some of the follow-up works. So these are some of the papers, and there's many more in the field now. But Nick Navin et al., that paper in 2011 was one of the first, the first paper I think that did single-cell sequencing to estimate subclonal, to detect subclonal heterogeneity among tumor cells. And that was a breakthrough that people can now experimentally do this, right? But it has taken basically 10 years to figure out how to deal with the sparsity of this data and how to get reliable measurements out of it. Why is this problem hard? So let me just sort of do a cartoon to show you what we're trying to get at. So in here, imagine that there are 3 subpopulations of cells in your tissue. The normal population that basically has 2 copies, one that's 2 different one the blue copy from the haplotype 1 from say the mother, and 1 copy, the red copy from the father. Like that's what we should have in every cell. But then let's say that there are 2 other subclones, one that has a loss of heterozygosity, so copy neutral loss of heterozygosity, which is very common in cancers, right, in this segment, and the other that might have an overlapping segment that is gain in the red copy, right? So maybe this is the underlying picture, but we don't see that. And what we actually see are basically reads that are sparsely sampled along the genome, a small set of reads per cell or per spot on a VCM array. And if the read overlaps with a SNP, then you will be able to see whether it is an A or B allele. If it doesn't overlap with a SNP, it is still informative. It tells you about, it sort of contributes to the counts that tells you how many total copy number there is, right? So you would have this across different cells, and the data sets now can range between a few hundred to maybe 10,000 cells, a few tens of thousands of cells, right? So, you see that we want to basically infer from the observed data what the underlying population structure is like. Okay, and I just want to reiterate the allele-specific aspect of this is really interesting. It turns out that you can actually discover, there's a lot of these kind of, for example, gene conversion events that you would not be able to detect if you were just to look at total copy number. So the challenges here is that the reads per cell, there's very few of them, and you don't know where the breakpoints are along the genome, right? And also you don't know the underlying phase. So you don't know how the different alleles are stringed together into one haplotype. And you need to know the phase in order to pull reads that are adjacent to sort of overcome the sparsity. Right. So the model we have is that, well, I'm just going to show you very quickly the model, and then we'll go on to what the model actually, reveal in the data. But what we have is that for each cell, we have reads that are supposed to be sampled from the cell from a Poisson process with an underlying rate function mu t, which is a background rate, and this delta it, which is a piecewise continuous rate function that has change points that are shared among a subset of the cells. Okay, and furthermore, if the read overlaps with the heterozygous site, then you have this you have an indicator of whether it carries the alternative reference allele, and you observe that indicator, but then it depends on this underlying phasing information, right? Whether the copy number changes on the major or minor haplotype. So if there's anything I want you to get out of this model, I think this kind of picture is really, really intuitive for me. So basically, if you look at a region on the genome, right, that, across cells, let's say this region, it's a small enough region that you can think it's homogeneous across a region, then basically you can plot out the normalized coverage of that region versus its major haplotype proportion. So A versus A plus B. Right. So if it's a diploid cell, so each cell could be a dot on here. If it's a diploid cell, the normalized coverage should be 1 because there's no change. And the major haplotype proportion should be 1/2 because it should be A and B, right? If there is a deletion, then it goes to let's say to 0.5 because it's 1 copy deletion, and then it loses copy number, so it loses the heterozygosity. So the major haplotype proportion will be either 1 or 0. If there is some amplification, then it goes from 1/2 to maybe 1.5 1/3 or 1.5 2/3, right? Depending on which haplotype is amplified. So basically, if you put a cell on this plot, it should belong to one of the canonical points if you have a homogeneous enough region, okay, where there's no breakpoints within the region. So that's what we're going to try to do. And that's essentially what these parameters give you. These parameters just tells you where it is on this plot. So let's look at an actual sample. So this was one of the samples, one of the first samples we looked at, actually. So this is a gastric tumor. It's a single cell DNA. So let's first start with DNA. That's the easiest, right? And then move to ATAC, then move to RNA. So this, if you look at its cell ranger, and so this is from 10x, so you have the cell ranger output, you see that there is amplifications of chromosome 7 and 8, and it looks to be subclonal. And then these are noisy cells, so you don't ignore that noise up here. And then there's this, so this is a male sample, so there's only one X chromosome, etc. Okay, so if you were to look at, say, chromosome 7, right? So that looks like a subclonal amplification. You indeed see this cluster here, that's the amplification, but you also see this mirrored subclone. So that was one of the sort of surprises we ran into, is just how many cases of such mirrored subclones we see in our data, which is, you wouldn't be able to see this if you did bulk sequencing, and you wouldn't be able to see this even if you did single cell sequencing but ignore the allele-specific aspect. So you see, for example, for this chromosome 8 amplification, you also see a major subclone plus points that if you do the normalization correctly and segmentation correctly, that basically falls on top of the canonical points. So basically this gives us a way to even just, from one region of the genome, identify 4 subclones in the tumor. Okay, so there's a lot of clonal heterogeneity in here that was previously masked. And here's again a deletion. You see that there's a major subclone, and there seems to be 2 cells here that has a deletion of the other haplotype. Okay, then they're sort of co-evolving in the tissue. So okay, so we applied our method because at that time we just wanted to know how much subclonal heterogeneity there is among single cells, within samples, because from the bulk sequencing world, we sort of have some ideas, but we didn't really have a clear picture of that. So we applied this tool to different cancers like colon cancer, gastric, breast cancer, and then I'm just showing you for each cancer sample 2 example regions that we extracted from that had either an amplification or a deletion from the bulk sequencing. And you see that you see amazing heterogeneity among the cells, that they are just really, for example, for this region, you have 1, 2, 3, 4, 5, 6, would seem at least 6 subclones. Okay, so of course I'm sweeping a lot of the technical details under the rug. So with that model, we have ways to estimate this, and then it's in a R package, the alleloscope R package, from which you can get these plots. Okay, so but what this gives us is an opportunity to study tumor evolution using single-cell DNA or single-cell ATAC sequencing data. I say ATAC because ATAC is sort of like DNA, except for you're looking at the open chromatin regions. And so it's much easier than say single-cell RNA sequencing data in terms of getting the copy number information out of it. So I can show you that for this sample, we have the DNA profiles for these regions, and we have the ATAC profiles. And you see that for this ATAC, it's usually sequenced at much lower coverage. So you see that the clusters are more dispersed. They're not as tight as the DNA, but with good statistical modeling, you can actually extract the cluster centers and the cell's clonal identity. So from that, we can build a clonal tree that relates these cells. And then also we can just identify differentially accessible regions in the ATAC that after conditioning out, after controlling for the underlying DNA copy number change. Okay, so basically what this, from the single cell studies, it basically highlights the amazing subclonal heterogeneity that is in these tumor samples that you are not able to identify previously using bulk sequencing. And that, that explains why tumor cancer is so dangerous, because they are so heterogeneous. And then, but I want to show you what you can do now when you transport these methods into the spatial world. Okay, so here is a study. It's not our study. It's a beautiful study that was published last year in Nature, where they sort of did this one of the pioneers in this type of analysis. So they were looking at a prostate cancer sample, and they applied this tool called Infer CNV on this is a VCM on the spots. And this on the top here, you see an Infer CNV output plot, which is basically x-axis. So this is a location along the genome, and then each row is a spot. And you have red supposedly when there's amplifications, and blue when there's deletions. So it sort of suggests that there's a lot of copy number changes among the tumor cells, as well as a lot of heterogeneity. So they clustered it into different clusters, A, B, C, D, E, F, G. And then when you color them in space, they are contiguous in space, right? So they're sort of smooth. which is what you expect, right? Because clones sort of expand smoothly in space. And then so you can see where the different clones are. And here the picture is, here is the pathologist annotation. So just looking at the H&E image that comes with it, they can annotate the sample. And then here is the sort of infer CNV analysis. And what was interesting here, so, oftentimes I'm asked a question like, from the spatial data, what are some of the analyses that you do, for example, counting the subclone density and things. This is all you can do in space in single cell, right? But what does spatial give you? Well, what we found, what they found, and what we're going to follow up on the study is that they found that in these regions of the tumor that the pathologist annotated as normal, that in fact they found a lot of copy number changes. So what this suggests is that surrounding the tumor in the supposedly benign regions, there's already a lot of genome instability of the same type as the ones as the tumor cells, which was kind of interesting, right? As that sort of, shows you how the cancers, not just the cancer cells themselves, but the surrounding areas probably inflamed and also going through changes. But they use infer CNV. Now, we're close to the broad, and infer CNV is a tool that was developed, but I'm going to be a little critical here. In infer CNV, it's a sort of a bottom-up smoothing-based approach, and you know that whenever you have a random sequence of numbers and you do a local smoothing, you're going to see ups and downs. That's just an artifact of the smoothing. You're going to see waves, right? So, they didn't really anchor their analysis on any matched bulk DNA. So here is, for example, the infer CNV analysis of a tumor sample. So you would think that these are gains and losses, but we did a single cell DNA sequencing experiment on this exact same sample. Now that is gold standard, and from there we know where the true copy number changes are. It's on 7 and 8 and 20, and there's a deletion on 21. So you see that indeed you get it right on some of these regions, but you get a lot of false positive segments, and then when you cluster based on them, it leads you to wonder, are you really finding clones in the, in terms of copy number clones, or are there, because they may be valid, legitimate clusters, but the question is, what is driving the clustering, right? So I'm going to just show you how we're going to try to do a better analysis where we can extract true copy number changes that, that reveal subclonal information. So to make, I'm going to go really fast here. So there are two insights in our method. One is that oftentimes you have, or it's easy to get bulk DNA sequencing on the same sample. Okay, so that's now very cheap, and you can just take, it doesn't take too much cells. And then you can use the bulk DNA sequencing data to sort of extract the segments that drive your clustering. And now for in the single cell or spot data, your job is not to do the segmentation. Your job is basically to assign the cells to the right copy number state at each segment. And that is much more stable, right? So then we developed basically a non-parametric clustering-based procedure that sort of has a nested dirichlet process that mimics this evolutionary process where you have copy number gains and losses happening, and then that basically, the technical details, ignoring the technical details, it basically gives you a clustering of this data that is anchored based on whole genome sequencing data. Now, we are often asked the question, what if you don't have whole genome sequencing? Because sometimes maybe for FFPE tissue, you can do whole genome sequencing, but maybe it's not good quality. So if you don't have whole genome sequencing data, we would, our method, what we recommend is to go through a top-down approach rather than a bottom-up smoothing approach. So you start with chromosome arms, and then apply the subclone detection, and then going into subclone, estimating an average profile, and then going into the subarm level. So a top-down segmentation approach. Okay, so here is an example of a slide DNA-seq clustering, right? So this is a tumor where they found 2 subclones, and then we were able to recover those 2 subclones. So this is sort of just a way to do a sanity check that we're getting the right profiles. And then again, a validation on 2 adjacent slices of the same squamous cell carcinoma sample. This is a public data set where I had a expert pathology annotation done on it, so we can compare our tumor versus normal annotation on this, and you see that we were able to get. This is just proof of concept, sanity check that we are able to get to the real biology. Okay, so now let's get back to the sample we started off on. Okay, so this is the colorectal metastasis, where on the left, you would expect if you were a pathologist, you would look at it and say, that's cancer right there on the left. If you were to do a deconvolution, say using RCTD or some other method, you will find that, okay, indeed, the left part is enriched for epithelial cells, as you would expect if it was cancer. So that's another sanity check. And then if you were to do clonal type clustering, you will find that there are 3 main clusters, orange, yellow, and blue. Now, so far, this method we have still don't, it's basically treats each spot like it's a cell. So that's a drawback. I mean, these are mixtures, right? So you would imagine that maybe these little messy clusters, these dark purple and cyan clusters, is because those are mixtures of normal and infiltrating fibroblasts and other macrophages. So let's ignore that for now, okay? So let's focus on the 2 main tumor subclusters. You see that indeed they have different copy number profiles. That is very evident if you were just plot out their copy number states. Okay, so I, in our paper online, we have many other examples and validations, but I just want to move on to the next subject, which is how do we think about these 2 subclones in terms of how they're interacting with their local spatial environment. Okay, so remember I said that there's a lot of infiltrating normal cells here, and then we're going to figure out what it's doing with the tumor. So this is work that is led by a very talented student, Kai-Shu, and Kai-Shu is actually sitting there, so you can ask him hard questions after this talk. We call this method niche DE expression. So I'll show you what that means. So what are we trying to get at here, right? So we have, let's say, a tumor, and we have this VCM data or some other spatial data, and let's say that we know through deconvolution or through cell type assignment what the different spots are. So some are normal, some are tumors, some are myeloid subspots, some are stromal. Okay, and then there are places where these spots sort of coexist, they co-localize, right? So there's now many different kinds of co-localization analyses where you can look at where, say, for example, macrophages and tumors sort of come together, right? But what we want to know is how this affects the genes, especially which genes in, say, macrophages become differentially expressed in the presence of, say, tumor cells in its niche. So that's the sort of question that we find that when we work with collaborators, we're repeatedly asking is how these niche effects, how these different spatial niches affect the gene expression. Okay, so in order to answer that question, we first have to define what is a niche, right? So the way that, there's nothing complex about this. So basically what we did was for each, we started from the single cell model, because you can think about niches, cell niches, in a much more natural way when you reduce things to cells, right? So for a given cell, you can think of its niche as a kernel smoothing of the cell type composition in its neighborhood, right? So for different, let's say that there's 3 different cell types in this data, purple, red, and green, 3 different cell types. Then you figure out within for each niche, for each cell, what is the concentration of red in this niche, and purple and green. And you can use different bandwidth to get different neighborhood sizes. Okay. And then for a single cell, in a single cell world where you can observe things at the resolution of single cells, then you would just have a very simple regression model where the RNA counts for that gene, that cell, would depend on what cell type it is from, so the mu-TCG, the cell type average expression, plus some a linear model on the niche concentration of the specific cell type, say cell type purple, and the beta is the parameter you're interested in. So beta tells you how the effect of additional cell type T prime in the niche of cell type TC. Okay, so that's at the single cell world, but we don't always have single cell level data. And even the, like Xenium and Mirrorfish, they only give you gene measurements for a subset of genes, right? So we would really like to have a way of getting at the niche effect when we have a spot level model. So when we have a spot level model, what it turns out that, it's just a very simple mathematical derivation that I'm not showing here. Basically, if you do this regression, you can estimate these parameters beta, which are essentially the same as these parameters beta in the single cell world. So that's the insight here. You can do a similar regression with these pesky PSTG terms that you can compute using your deconvolution that ends up giving you the same exact betas that quantify the niche effect as if you saw the single cell data. Okay, so that just reduces down to another regression. And what's more is that, oftentimes cells co-localize. So, macrophages and fibroblasts often co-localize in tumors. So oftentimes it's hard to tease apart the contributions of, say, macrophages and fibroblasts together, each separate effects on a gene. So when you have a lot of this co-localization, then what you would like to do is a hierarchical testing procedure, where for each gene, you just figure out whether it has some kind of local expression, okay, in a given cell type. Say you do a first high-level test whether the gene is niche expressed in, say, this cell type. And then you go down to a second level where you look at the specific cell type that is causing this niche expression. Okay, so this turns out to give you more power to detect true niche differential expressed genes. Okay, so in the end, basically, you get to say something like gene 2 is cell type, it's macrophage niche differentially expressed when fibroblast is enriched in niche. Okay, so I'm going, due to lack of time, I'm not going to have time to talk about ligand receptor signaling, which is something we're excited about right now. But, basically, if you had a whole transcriptome data and identify all the niche differential expressed genes for a given cell type within the presence of another cell type, then you can start looking at the ligand and receptor expressions of these cell types to figure out whether there is ligand receptor signaling, because the differential expressed genes will tell you whether downstream pathways are activated. So that's something that we can do now, and it's still being we're still working on this, in fact. So okay, so so let's go back to this example that we started with, right? So remember that we identified two different two big subclones, so that we ignored some of the smaller subclones for now because there's not enough cells really to drive the power of niche differential expression analysis. So let's focus on the orange and blue subclones, right? And then in fact, let's go back and look at the H&E. So if you were, so we worked with a pathologist who really understands this, and he taught me that, if you look at the H&E, there's a lot of white stuff here, which means that there's more demosplasia, so more collagen formation, more fibroblast infiltration, and on the top part there's less of that, and then the bottom part, the tumor cells are more differentiated and they're less differentiated on top. So okay, is that true? If you were to look at a deconvolution, so this is an RCTD deconvolution estimating the stromal cell proportion in the spots, you do indeed see more fibroblast infiltration at the bottom part. Okay. So so I'm masking a lot of details, but we apply niche DE to this sample to look at what is different between the fibroblasts at the bottom part of the picture and the fibroblasts. So there's you have enough cells. You have a handful of cells that or spots that contain fibroblasts on top as well. So we asked, what is the difference between these two subclasses of fibroblasts? So basically, we perform niche DE in fibroblasts with this, differential between subclone 1 and subclone 2. So we found that we found a list of niche DE genes in each, and then we found that there's actually enrichment of interferon signaling at the bottom subclone. So it's interferon signaling between subclone to these cancer-associated fibroblasts at the bottom, whereas on top is a different sort of dynamic that there's Notch1 signaling and fibroblast activation instead of this. So I won't pretend that I understand more about the biology here, but this is it's interesting to my collaborators on this, and then this would be something that we may want to follow up on. Okay, so that's it. I skipped over a lot of the technical details and and validations and power studies and things like that. But if you're interested, you can take a look at our papers on this, and we are still in the process of polishing the software. But if you're interested, you can download it from here and take a test and let us know how it goes. We would love to work with you to apply this onto your data. And then finally, I just want to thank the students and postdocs who worked on this. So the first part was led by Jojo and Jiyun here, and then Kai Shu led the second part on HDE. I want to also thank our collaborators in Hanli's lab at Stanford, and also, of course, our funding. Thanks.
Questions for Dr. Zhang? Maybe it might be better to share your voice.
Hello, very interesting work. I would like to know how does the niche be compared to the wet lab techniques to describe cell-cell interactions like lipstick?
Can you repeat?
Yeah, like how does the results from the niche compare to the wet lab techniques for cell-cell interaction like lipstick?
We would love to try to validate it using these wet lab techniques, but we haven't gotten the resources to do so yet. We would love to give that a try.
So there's a question from online. Can we use niche DE for common and complex diseases like cardiovascular disorders to perform differential expression analysis and cell deconvolution?
Okay, so that's a good question. Niche DE is not a deconvolution method. It is a method that builds on deconvolution results. So if you have spot level data, you need to first do deconvolution to get a cell type proportions in each spot, and that is what we input into niche DE. And the accuracy, starting with a good deconvolution in the beginning is very critical. So we are relying on the people who are working on these deconvolution methods. And yes, we are applying it to other diseases. For example, we're trying to use it to analyze a kidney fibrosis data set where the concept is similar. You have different cell types and you want to know how their co-localization is affecting the gene expression.
Hi. So it looks like here you mostly showed the HDE on this one set of cells. Are there any significant differences for applying it for like multiple, I guess, multiple spatial images from multiple people and so on?
Oh yeah, good question as well. So I'm showing you this just simple example here, but in the bioRxiv preprint, we applied it to a set of samples, which usually you would want to do that to get more data points and to power your analyses, right? The more spots you have, the better. When you apply it to different across samples, then you have to deal with also like annoying things like batch effects. Well, I think just what are the batch effects and how to deal with them in spatial data is itself right now, I think, an unresolved issue. I'm still trying to grapple with it, like learn for different kinds of spatial technologies, what are the batch effects. But one simple hack that we have tried was basically in the regression, there's a regression and you can add the batch indicator, and that will at least adjust for the mean level differences in the batch. Okay, so it may not adjust for everything, but at least it gives you a better peace of mind than just applying without it. So basically putting a batch correction term here. Mhm.
You were mentioning there about you run the deconvolution before running niche DE, so it's more designed for doing the 10x Visium data. For something like Merscope, can you still apply it?
Yeah, so we in the preprint we have examples applying it to cosmic data and seekfish. I think certainly it can be applied to Merscope. Is easier because then it reduces to the top equation here rather than the bottom equation. So directly you can, it's basically a regression. It's still good to do the hierarchical FDR because it sort of reduces the number of tests you make. You can try. I think we're still in the process of trying on different technologies to see how exactly it works.
It's really interesting. So this is for like spatial data. So in this equation, I'm not sure where the spatial coordinates would come in.
It doesn't. Oh, you mean in here? Yeah. Oh, yeah. So it comes into this term NS T prime, where NS is a spot, and T prime is a cell type. So that is the concentration of the cell type around the spot. And we show that, of course, it depends on your, I skipped over this, but it depends on your kernel size. So of course, if you increase the bandwidth, you're going to be looking over a larger niche area, larger neighborhood. So what we found is that in order to have sensitivity for signals across different bandwidth, you just need to have like a 3 different bandwidth. I mean, it's not as, for example, if you have a small bandwidth, it will be sensitive for all the local interactions, and then a medium bandwidth and a large bandwidth. You don't need to have too fine a grid in order to have space sensitivity across the spectrum.
Yeah, I was wondering if the subclone definition changes, how difficult is it to repeat the deconvolution and repeat the spatial analysis you described later?
If the subclone, if you have different subclone identity.
Subclone definition changes, or maybe you want to compare two competing models of the subclone.
Yeah, so you would just apply this to one model and another model and compare them. I mean, right, that's why I sort of didn't go over those details, but what we did was we used deconvolution to estimate the proportions of all the normal cell types, like fibroblasts and macrophages and things like that. Whereas for the tumors, the part that's labeled epithelial, we use the copy number-based method to identify the subclones, right? So your normal cell type proportion wouldn't change because that's separate. And then your subclone, if you try different subclone detection methods, it might give you different things. Hopefully not, but it might give you different things. Then you would just plug them into the method to see what you get. Yeah.
For these multiple bandwidths, do you think there's any merit in putting all 3 of these in the same model?
That's what we do. We basically we have a default bandwidth, and we use a Cauchy combination test to combine the signal across different bandwidths.
Following up on that question, it's an amazing presentation, by the way. Thank you. Can you tell from which bandwidth some genes are more differentially expressed or not? Feels for like a ligand receptor analysis, it would be very different. What type of communication will be happening across the 3 different kernels? Yeah,
that's a really intriguing point, and that's something that we would like to know. Whether it's different bandwidth or, and we started along this path. We plotted what's the optimal bandwidth, like what's the likelihood maximizing bandwidth, right, for different genes, right, and then for different ligands, receptor pairs, what are the downstream genes, what are their maximal bandwidth. We saw some patterns, but we don't know what's the truth. So I have to say that we're just grappling in the dark on that. But that would be certainly something very interesting to study.
Any questions for Dr. Zhang? I have one. So I didn't fully grasp until just a second ago that the identification of tumor or not is being copy number driven. Does that mean that if you have a tumor that's driven by a fusion or that has a very that tends to have a normal karyotype, that the method's going to fail to detect any sort of niche effect for it?
Oh yeah, certainly. So this method, well, the subclone detection methods I mentioned, they are limited to only finding subclones or tumor cells that have copy number changes, and that's a lot of tumor are like that. But certainly, we now know that there are single nucleotide or gene fusion or even epigenetic driven subclones, right? So there might be. The thing is, at the single cell or spot level, it is so hard to detect those. So yes, we're missing a lot, I think. But copy number is one of the, because for these events, you can just pull, you can sort of pull across reads to overcome the sparsity. But certainly, they're probably only a small fraction of what you might expect.
Sorry, that's one more question. So you mentioned the Erickson paper, the prostate cancer paper, and you referred to where they were seeing effects in the normal cells or the non-tumor cells.
Yeah.
Thought that might be artifacts of methods that were being used.
Oh.
Did you see less of those in your analysis?
No, I didn't mean to. No, I mean right. So that's actually real, I think, and they actually validated it with qPCR. So so those are right. So this one, right? So you're talking about this one. So they found this intriguing, this yellow cluster up here, which has copy number changes, and it is in a region where the pathologists say is benign. We actually applied our methods to this, and we found the same thing. And then so, okay, not that we didn't believe it before, because they provided other evidence that it is real. But this just shows why looking at these subclones in space and comparing it to pathology annotation can yield more insights. Because I think this was, at least from I don't know if I missed any literature before, but this is the first example where you concretely see for the slice, here are the cells that look benign, and then they actually contain these large copy number changes that are in the adjacent tumor region. So that just opens up a bunch of questions as to how this, what's going on for this cancer. So it's just like, I like to use that as an example that this is what spatial genomics can give us.
Any more questions for Dr. Zhang? Thank you very much.